This video describes the laboratory procedures and workflow for detection of SARS-CoV-2 directly from saliva using the Optigene Limited COVID-19 Direct RT LAMP kit. The assay is carried out manually and is currently being used to process 1,500 to 2,000 samples per day. The procedure and workflow was developed at the Animal and Plant Health Agency in collaboration with colleagues at the University of Southampton, Optigene, Hampshire Hospitals Foundation Trust and Dr. Veronica Fowler. The overall project leader was Professor Keith Godfrey from the University of Southampton. In this SARS-CoV-2 assay, viral nucleic acid present in a saliva sample is amplified and detected with the loop-mediated isothermal amplification or LAMP technique carried out using a Genie HT instrument from Optigene Limited. The method involves the reverse transcription and subsequent amplification at a constant temperature of 65 degrees C of a specific viral target sequence which are detected in real time by the fluorescence emission of a DNA intercalating dye. Following amplification, the Genie HT machine automatically runs and generates an annealing curve where the reaction is heated then slowly cooled. This acts as a secondary confirmatory check, ensuring LAMP amplicons are specific to SARS-CoV-2. The instrument integrates the results from both the amplification and anneal steps and reports the sample as positive or negative. Because the direct LAMP assay takes approximately 20 minutes and does not require RNA purification, it offers the potential of a rapid test for mass screening. An outline of the assay is as follows. 50 microlitres of saliva is mixed with 50 microlitres of lysis buffer. This is then heated at 98 degrees for 2 minutes. 5 microlitres of the resulting sample is then added to 20 microlitres of LAMP master mix prior to the LAMP reaction being carried out on the Genie HT instrument. This video has been developed to describe the practical steps in carrying out testing manually. The practical procedure for testing is divided into 3 key steps. Step 1, preparation of reagents and materials prior to testing. Step 2, sample reception, LIMS registration and tube racking. And Step 3, direct RT LAMP assay. Step 1, preparation of reagents and materials prior to testing. PCR plates with rapidized sample lysis buffer and Genie tube strips containing direct RT LAMP master mix can be prepared in advance and frozen for later use. Ready-made plates and strips can be safely kept at -20 degrees for up to 7 days. Alternatively, these reagents can be prepared for immediate use as required, but we have found that this approach simplifies testing significantly. To prepare 96 well plates in advance, pipette 50 microlitres of the rapidized sample lysis buffer into each well of the first 11 columns of a 96 well PCR plate and cover with PCR sealing film. Freeze the sealed plate at -20 degrees. Prepare the Genie tube strips containing LAMP master mix by pipetting 8750 microlitres of direct RT LAMP master mix into a suitable sterile plastic tube. Add 1250 microlitres of 10 times COVID-19 primer mix and mix well. Keep on ice at all times. This volume of reaction mix is enough for approximately 500 individual LAMP reactions. Aliquot 20 microlitres of the prepared reaction mix into each tube of the required Genie tube strips. Loosely close the Genie tube lids to the first position so that the lids remain closed but not locked down, and store them supported in microtiter plates, 6 Genie strips per microtiter plate. All preparation and dispensing steps must be carried out on ice. Freeze the dispense strips at -20 degrees until required. Step 2, sample reception, LIMS registration and racking of universal tubes. Saliva samples arriving in 30 ml universal tubes must be sorted and racked in a dedicated sample reception area separate from the testing laboratory. The samples are registered into a LIMS, Laboratory Information Management System, by scanning the barcodes on the universals. These are then arranged in racks in groups of 88 universals made up of 11 columns of 8 universals, ultimately corresponding to 11 Genie tube strips of 8 samples. For 2,000 samples a day, this corresponds to 23 racks of 88 samples. For each rack of samples, LIMS generates 12 sample sheets, which will be used later to register the details of each group of 8 samples in a single Genie tube strip onto the Genie HT lamp instrument. The sheets include a master sample sheet with plate reference number, which mirrors the arrangement and order of the 88 samples in the rack, 11 columns of 8 samples, and 11 Genie tube strip sample sheets with a barcode, one for each group of 8 samples. Rack samples, together with their corresponding sample sheets, are transported to the laboratory in plastic clip lock boxes, or alternatively, paperwork can be printed in the testing laboratory from LIMS using the plate reference number. Step 3. Direct RT lamp assay. The lamp assay is divided into 3 parts. Ideally, each part of the process should be carried out in a separate laboratory to minimise the chances of cross-contamination, following established molecular biology practice. In the first part, the saliva samples are mixed with lysis buffer to inactivate the virus. This is performed in a Class 1 or Class 2 biological safety cabinet. For each rack of 88 sample universals, thaw 1 pre-prepared 96-well PCR plate preloaded with rapid lyse buffer. For processing, the saliva samples must be transferred from the individual universals into a 96-well plate containing the rapid lysis buffer. To ensure accuracy in the distribution of samples, a buddy system is used. 2 people work together to process each separate rack of samples at one cabinet. One person, the operator, checks that the orientation and order of samples in the rack matches the master sample sheet. The operator must call out the final 5 numbers of the barcode on each sample tube. The buddy then checks this against the ID provided on the master sample sheet and verbally confirms the barcode. The buddy then dictates which well of the PCR plate the sample must be added to. Following this system and using a 200 microlitre pipette, 50 microlitres of saliva from each sample is transferred to the corresponding well of the 96-well PCR plate. Use the master sample sheet to dictate the specific location of each sample within the 96-well PCR plate. For example, sample 1 to well A1. The sample is mixed with the lysis buffer by pipetting up and down 5 times. The pipette tip is discarded after this step. To avoid cross-contamination, avoid touching the inside of the universal with the pipette barrel. Once all 88 samples have been transferred to the PCR plate, seal the plate with a foil PCR plate sealer, spray the plate with 70% ethanol and incubate for 1 minute before removing the plate from the safety cabinet. Using a PCR thermocycler, heat the PCR plate to 98 degrees for 2 minutes. A cooling step is added to the PCR program to reduce the sample temperature to 20 degrees. Plates can be removed from the PCR machine once the temperature has reached 20 degrees. The second part of the lamp assay involves addition of the sample lysis buffer to Genie tube strips containing the lamp master mix and is performed either inside a Class 2 biological safety cabinet or under a laminar flow PCR hood. First, for each 96-well PCR plate of samples to be processed, remove 12 Genie tube strips containing master mix from -20 degrees storage. Once thawed, keep the strips on ice until immediately before use. To ensure that samples are loaded onto a Genie strip in the correct order, orientate the opening hinge and lid on the right-hand side of the strip. This orientates tube 1 at the top of the strip with samples 1 to 8 loaded down the strip in order. Using a 1 to 10 8-channel multi-channel pipette, pierce the foil plate seal of the PCR plate and transfer 5 microlitres of sample lysis buffer mix from each column of the PCR plate into one Genie tube strip containing 20 microlitres of master mix. The appropriate localisation of samples in this strip is critical. The samples in one column of the PCR plate are added to one Genie tube strip. Sample placement on each Genie tube strip should mirror the Genie tube strip sample sheets. Importantly, once samples have been transferred, firmly press the lids of the Genie tube strips down, ensuring the lids are pushed into the second lock position. If any lids look damaged or aren't closing properly, discard the strip and start again with a fresh strip. Once all the samples have been loaded, reseal the plate with an additional foil seal and store at 4 degrees for potential further use or disposal. Following completion of the 11 sample strips from each rack of 88 samples, prepare a negative control strip. To do this, add 5 microlitres of DEPC treated water to each well of a Genie tube strip containing master mix in the same environment that the sample Genie tube strips were prepared. The use of a negative control strip every 11 strips is essential, and checking for amplification in these strips during subsequent lamp reactions is an essential early warning of potential contamination. Mix by vigorously flicking the Genie tube strips multiple times. Hold one end and flick, hold the other end and flick. Centrifuge the Genie tube strip in a Genie centrifuge for 10 seconds to settle the contents of the tube and ensure no bubbles are present at the base of the wells. The third part of the lamp assay involves the actual amplification run on the Genie HT instrument. Transfer each Genie tube strip to a Genie HT instrument. If instruments are in a different laboratory, transport the ready prepared strips on ice. Load the Genie tube strip into an empty heat block on a Genie HT instrument. Select the COVID-19 Saliva Direct Plus protocol on the startup menu and ensure that only the appropriate heat block is chosen. Press the sample box on the touchscreen of the Genie HT instrument. Tap the text box and scan the barcode from the sample sheet of the Genie tube strip being tested. This transfers the sample barcode details of each individual sample to the Genie HT instrument. Ensure no other blocks are selected at the same time. Tap to deselect any unnecessary blocks and press play to start the COVID-19 Direct Plus protocol. The negative control strips are run on the Genie HT instrument using the COVID-19 Direct Plus protocol but with no barcode scanned. Following completion of the run, including the cooling period which forms part of the COVID-19 Direct Plus protocol, the completed Genie strips can be removed and disposed of. Before removal, press the lids of the Genie tube strips to ensure the lids have not opened slightly during the lamp process. Do not remove the Genie tube strips until the temperature has cooled to 30 degrees. Remove the strips gently to try and avoid opening the lids of the Genie tube strips, making sure to carefully hold both ends of the strips to minimise bending. Dispose of the completed strips regularly. Check the results of the completed lamp reactions. If any samples are positives, inform the lab supervisor and do not remove the strip. Working together with the supervisor, check the lids of the Genie tube strips prior to removal of the strip from the instrument. This is to reduce the chances of amplicon from positive reactions contaminating the laboratory. The lab supervisor can then check for amplicon contamination using the negative control strips. A control Genie tube strip is run at 3 points during the day. At the beginning of the day, at midday time point and at the end of the day. These 3 positive-negative control strips are run on all Genie HT instruments being used for testing. The control strips are set up as detailed in limbs. Tube 1 positive control, Tube 2 negative control, Tubes 3 to 6 blank, master mix only, Tube 7 positive control and Tube 8 negative control. 5 microlitres of DEPC treated water is added to negative control reactions. This is carried out in the biological safety cabinet or lab in a flow PCR hood where the test samples were added to the Genie tube strips. 5 microlitres of positive control is added to positive controls at the last stage of preparing a control strip in a Class 1 or Class 2 biological safety cabinet. The control strips are used by LIMS to validate the lamp reactions tested between different control strips. Positive and negative controls are prepared in advance as single-use aliquots. Positive control material is any material which reliably amplifies to produce a positive result. Possibilities include inactivated SARS-CoV-2 virus or the synthetic control produced by Optigene. Following completion of the lamp run, the resulting lamp test result data is automatically picked up by the LIMS system from the Genie HT instruments, allowing result checking and authorization remotely via LIMS.